Imagine a secret superpower, not, from a comic book, but hidden right there in your own DNA. A silent protector, passed down through countless generations, from ancient relatives you've never even met. What if that ancient inheritance from beings that walked the earth hundreds of thousands of years ago is actually helping you fight off diseases today? Get ready, because we're going on a deep dive into the fascinating world of the Denisovans. These mysterious human relatives, they once roamed vast stretches of Asia, and groundbreaking new research is connecting their ancient environments to a surprising aspect of our modern health, our resistance to infectious diseases. So the big question we're exploring is this. How do the challenges Denisovans face, the pathogens, the climates, the very disease vectors in their ancient habitats, how did that leave such a profound imprint on our genes? An imprint that it still matters to you right now. Our mission today is to unpack a single, really illuminating study. It used advanced geospatial techniques, intricate climate modeling, and cutting-edge genetic analysis to reveal how Denisovan habitats sculpted modern human disease resistance. Think of it as maybe a shortcut to understanding a massive and truly surprising evolutionary puzzle. So let's start at the beginning of our journey. Let's picture where these Denisovans actually lived. The study focused on 3 key archaeological sites, and what's truly striking is just how incredibly diverse their ancient climates were. It wasn't like a single type of home, but a whole spectrum of environments. Okay, our first stop is Denisova Cave in southern Siberia. When you picture this place, think harsh, unforgiving winters and huge dramatic temperature swings. It was a subarctic environment, characterized by cold summers and no real dry season, sort of nestled within a boreal forest biome.
Right, so essentially dense pine forests, incredibly cold winters, and these short, cool summers. Not exactly a tropical paradise, as you say.
Then we climb much higher, to the Baishia Karst Cave on the Tibetan Plateau. We're talking about, what, 3,280 meters above sea level?
That high, yes. A really challenging altitude.
And this site experienced a monsoon-influenced subarctic climate. So cold, dry winters and cool summers. Also a boreal biome, similar to Siberia in its chill.
But with that added intensity of high altitude, and the study highlights, the paleo environment here was probably even harsher during their occupation.
Now, completely shift your mental image for the third site. Tamgu Haltu Cave, famously known as Cobra Cave in Laos.
A very different picture.
Totally. This was a distinctly different world. A monsoon-influenced humid subtropical climate, dry winters, hot summers, situated in a lush tropical seasonal forest biome. We're talking high annual precipitation and very little temperature seasonality, so consistently warm and wet. So you've got these ancient human relatives spread across incredibly varied landscapes, from what sounds like, frozen tundras and high-altitude plateaus
Right.
to humid, almost jungle-like forests. Now, here's a crucial piece of the puzzle. Modern human populations with the highest Denisovan ancestry today, they mostly live in tropical climates.
Interesting.
So this naturally begs an important question for you. How would adaptations from these cold, unforgiving northern environments actually help people thrive in the heat and humidity of the tropics?
That's a perceptive question, and it's absolutely critical to understanding the study's findings. The researchers' ability to reconstruct these past environments as well, it's what makes this research so powerful. And importantly, these reconstructions aren't just based on climate models. They're confirmed by tangible evidence, like faunal and pollen records.
So real physical proof.
Exactly. For instance, in Denisova Cave, they found remains of steppe-dwelling animals alongside those from subpolar forests. It indicates a rich, diverse environment. In Baishia, the evidence pointed to more of a mosaic of forest and shrub. But then, in Cobra Cave, they uncovered signs of a canopied temperate woodland, even remnants of arboreal species like giant pandas and orangutans.
Wow, pandas and orangutans. That really paints a picture.
It does. This kind of direct evidence gives us a unique, concrete window into the very specific environmental pressures these different Denisovan groups would have faced. And it deepens the mystery of that tropical connection you just highlighted.
And here's where it gets truly fascinating. Once we knew where they lived, the research modeled what kind of invisible threats might have been present. The diseases, the pathogens, and the carriers of disease, the vectors.
The local challenges.
Exactly. It's like reconstructing an ancient microbial battlefield. So in the cold north, at Denisova Cave and Baishia Karst Cave, the model showed high climatic suitability for Ixodes persiccatus.
That's the taiga tick.
Right, the taiga tick. And with these ticks comes a whole host of serious tick-borne diseases, like Lyme borreliosis, which you might be familiar with.
Definitely.
But also something called the Far Eastern PBE virus. TBE stands for tick-borne encephalitis.
And nasty brain inflammation.
Yeah, and it's still prevalent in South Siberia today and apparently has a higher lethality rate than its Western counterparts. These colder regions also showed moderate suitability for Plasmodium vivax.
A form of malaria.
Yeah, malaria.
Yeah.
And visceral leishmaniasis, which is a severe parasitic disease affecting internal organs.
So even in the cold, some tropical type diseases were possibly around, maybe less common.
Right. While marginal, these areas were still permissive enough for some transmission. In fact, they found ground squirrels in Denisova Cave, which are known carriers for zoonotic cutaneous leishmaniasis. That's a skin infection.
Interesting connection there.
But here's a critical point. These northern sites showed very low suitability for the Asian tiger mosquito, Aedes albopictus, and Nipah henipavirus. Probably just too cold.
Temperature limitations make sense.
And one crucial detail, Ascaris lumbricoides, a type of geohelminth that's a soil-borne parasitic worm, was likely absent.
Why is that?
Because its eggs need warm soil temperatures above 12 degrees Celsius to develop, and conditions were simply not met in these subarctic areas. Okay, now for a stark contrast, let's look at Cobra Cave in Laos.
The tropical site.
Exactly. This place was, well, a bustling hotspot of potential pathogens. It showed maximum or near maximum suitability for almost all the diseases and vectors they looked at.
Wow, really? Maximum?
Yeah, including P. vivax malaria, Ascaris lumbricoides, and this is key, the mean temperature of the coldest quarter was 15 degrees Celsius.
Above that 12-degree threshold.
Right, so it supported perennial geohelminth cycles, meaning these parasitic worms could thrive year-round.
Constant threat.
Constant threat. Also, visceral leishmaniasis, the Asian tiger mosquito, even the taiga tick, Ixodes persiccatus, somehow.
Even in the tropics.
Yeah, and various Asian Anopheles mosquitoes. They were all perfectly at home there.
So a much richer pathogen landscape.
Totally. And not just pathogens. It was also likely replete with toxic plant and animal species, like the poisonous milkweed Calotropis gigantea and venomous Niger snakes.
Good grief. A truly challenging place to survive. This striking variation, it tells a powerful story, doesn't it?
It really does.
The Denisovans inhabiting Cobra Cave were living in a warm, pathogen-rich environment, optimal conditions for disease transmission. They were facing a very different and frankly far broader set of zoonotic exposure risks and adaptive immune pressures compared to their relatives up in Siberia or Tibet.
Yeah, a whole different ballgame.
This ecological divergence in pathogen exposure is, well, it's truly profound. It really makes you wonder, given these vastly different environmental threats, what distinct immune defenses did they develop in response?
And crucially, how might those defenses have been passed on to us?
Right.
So what does this all mean for us? It turns out, when Homo sapiens interbred with the Denisovans, we didn't just get some, extra DNA.
It wasn't just filler.
Not at all. We likely acquired specific genetic adaptations alleles that were incredibly beneficial for adapting to new environments, especially the immune challenges posed by local pathogens.
Like inheriting a suite of ancient survival tools.
Exactly. Among these critical Denisovan-derived immune genes are toll-like receptors, specifically TLR1, TLR6, and TLR10, along with OAS1.
TLRs and OAS1.
Okay. These genes are involved in our innate antiviral responses, our body's very first line of defense. And they show clear signs of positive selection, meaning they were highly advantageous.
Right. So OAS gene variants, for instance, they influence our sensitivity to RNA viruses.
Yeah, essentially these genes contributed to increased resistance against a whole range of threats.
Yeah.
Bacteria, fungi, parasites.
Yeah.
Pretty useful stuff. Then there's an allele known as T10AA, I207. That's one originated from Denisovans.
Okay.
This allele enhances immune responses. However, here's where evolution's tricky balancing act comes in.
Uh-oh. There's always a catch, isn't there?
Sometimes. It also partially reduces the inhibitory effect of a protein called A20 on immune activation. Think of A20 as like a dimmer switch for your immune system.
Okay, it keeps things in check.
Right. So while reducing its effect boosts pathogen defense, it could also, perhaps in some cases, increase the risk of autoimmune diseases.
The double-edged sword.
Exactly. But the fact that this allele is highly frequent in populations east of the Wallace Line, that's a major biogeographical boundary, strongly suggests it was incredibly advantageous.
Especially in those newly encountered tropical environments with unfamiliar pathogens, the benefit outweighed the potential risk.
Precisely. despite the trade-off.
Right.
And let's not forget the cytochrome P450 enzymes, or CYP450.
Oh, yes, the detox enzyme.
Right. These unique variants, found in Denisovans, likely acted as ancient biological filters, helping our ancient relatives detoxify environmental toxins.
From those biodiverse environments like the toxic plants and venomous animals you mentioned in Southeast Asia?
Exactly. And they also seem to play a dual role in infection-related immune responses, contributing to the fight against vector-borne diseases like malaria.
So they gave them a leg up on both chemical and biological threats. Very efficient.
Seems like it.
This whole genetic mosaic, it truly highlights a critical balancing act for you, the listener. These ancient adaptations, they confer strong pathogen defense, which was undoubtedly life-saving for Denisovans way back when.
Makes sense.
But, as you mentioned, this often comes at the cost of a potentially heightened risk for autoimmune diseases in modern humans. It reflects a deep evolutionary compromise that precisely shaped our immune system. It made it robust against the unique array of pathogens Denisovans encountered.
Particularly those tropical ones, it seems.
Yes, but also leaving us with certain vulnerabilities that echo through our modern health challenges. It's a powerful testament to how deeply our history impacts our present biology.
And here's the truly groundbreaking revelation from this deep dive, the aha moment, if you will.
Okay, I'm ready.
The study connects all these dots to explain which Denisovans likely gave us these protective genes. This is the crucial piece that ties everything together. So we know that modern populations with the highest Denisovan genetic legacy, particularly in Melanesia, where people can have up to, 4.5 to 5.5% Denisovan DNA?
That's right, significant amounts.
They show a high present-day climatic suitability for tropical diseases, like Ascaris lumbricoides, those soil worms, again, Plasmodium vivax malaria, and Nipah virus.
Okay, consistent with a tropical legacy.
But here's the kicker, the crucial disconnect. These same regions have low climatic suitability for Ixodes persilicatus and Lyme borreliosis. The disease is prevalent where the Siberian Denisovans lived.
Ah, so the pattern doesn't match the northern Denisovan pathogen profile.
Exactly. This pattern is incredibly telling. It strongly suggests that the advantageous alleles protecting modern humans from tropical diseases like Ascariasis and mosquito-borne illnesses.
The ones found at high frequency today.
Right. They came from Denisovans, who inhabited the warm, humid continental climates, exactly like those found around Cobra Cave in Laos.
And not from their relatives who lived in the colder northern regions where ticks and Lyme disease were the bigger threats.
It's a very specific evolutionary gift from a very specific group of Denisovans adapted to a specific environment.
This is just a powerful demonstration of pathogen-mediated selection in action. It's evolution driven by disease pressure. As the study's author, Attila Trájer, put it so clearly, the extensive Denisovan genetic legacy related to pathogen resistance in modern humans appears less surprising. Denisovans inhabited highly diverse environments as reflected in the environmental reconstructions of even the relatively few known sites.
So the diversity of their homes led to diverse adaptations.
Precisely. The distinct disease profiles of their diverse habitats directly shaped our modern human disease resistance. It's not some general adaptation, but a highly specific one, finely tuned to the biological challenges of their tropical homes.
So you've just explored how deep into our evolutionary past our health connections really run. The distinct environments of ancient Denisovans, particularly those who thrived in tropical regions like Laos, directly contributed to the genetic tools you carry today to fight off infections. It's a powerful reminder, isn't it? Parts of your immunity against certain tropical diseases might have originated from an ancient interbreeding event.
With an ancient hominin in a warm, humid cave thousands and thousands of years ago.
It truly changes how you think about your own genetic blueprint. And understanding this link between archaic human habitats and modern human genetics, well, it isn't just a fascinating historical footnote.
No, it has real-world implications.
It can actually inform the development of new treatments and preventative strategies, especially for populations in regions with high rates of infectious diseases today.
It's ancient history informing modern medicine, potentially offering new pathways to health.
So as you go about your day, maybe consider this. What other ghosts in our genes might be silently shaping your life, waiting to be uncovered from the forgotten chapters of human history? The deep dive continues.